I'm Dr. Milescu. I am adjunct professor at Daytona State College-Deland campus, and today's video is all about brain dissection. We're going to identify the major parts of the brain. This here is a sheep brain. I'm going to show you how to dissect it today for anatomy lab, and also identify structures that you will need to know for your practical. All right, so the first thing you're going to need is a scalpel with a number 10 blade, and you have forceps, and then we have some probes here that we can use to be able to identify structures. So the first things first, I'm going to show you, this is a sheep brain, and it is the left and the right cerebral hemisphere here that you can see. And we have this as well, and this is the longitudinal fissure. And this is why we dissect a sheep brain, because it is quite similar. So there's the longitudinal fissure, if you can see that, okay? And then the brain tissue is convoluted, and we call that the gyri, gyrus for singular, gyri for plural. And then the minor, smaller grooves are called sulci. So sulci is plural. One groove is called sulcus. All right, so basically the brain, naming the regions of the brain is just like naming the skull. So the frontal bone, underneath the skull, you have this frontal lobe. So you have the left frontal lobe and the right frontal lobe. Left frontal lobe, right frontal lobe. Then you have the parietal lobe here. You'll have the prefrontal cortex. You'll have the somatosensory region here. The frontal cortex of the cerebrum is all about the seat of highest intelligence, your critical thinking skills, your judgment, all that is your personality, all that is frontal lobe. Parietal is more about somatosensory and movement, and then back here is occipital. So occipital is all about vision, interpreting what you see, and then here on the side is temporal. All right, so let's bring it close. Right here is temporal. Now the structure that you see back here, that would be the cerebellum, and you could see the spinal cord coming out. Now what I'm going to do is I'm going to stand up so you can see a little bit closer everything here. I'm going to turn it this way so you can see. Now I'm going to use my probe to show you. This is the olfactory bulb, as you can see right there. Olfactory bulb on both sides. Lifting it up, there it is. Olfactory bulb. All right, next you should see, take a look at the optic chiasm right here. Okay, optic chiasm. All right, so the optic nerve meets, so if it's coming from the left eyeball, it's going to that right crisscross to the right fibers of the optic chiasm. And then the right eye to the left eye to the right, crisscrossing at the optic chiasm. All right, then you're going to look for the mammillary bodies, and it should be right here. You can see the mammillary bodies right there, two little enlargements, okay? Oculomotor nerve should be right behind that, and I can't see the pituitary gland. I cannot see it. But what I can see is the midbrain. This here, that little bulge right there, that's the pons. And then here is the medulla. And wait, hold on. Let me make sure because I'm looking at a video. So midbrain, pons, medulla, and then spinal cord. Okay, so the really large enlargement here, pons, okay? What else? That's pretty much it that you can see on this side. Now what I'm going to do is I'm going to cut with a sagittal cut along the longitudinal fissure through that cerebellum. So let's see. I'm going to sit down. It's a little bit harder to do the video when you're dissecting because I have to bring it all the way up to the camera. So I don't know if you can see this, but you will be doing this in lab. So you use the scalpel like a pencil. You want to hold on to the brain. You don't want to cut up here in the air. So really just kind of tag it so you don't cut yourself. And do one nice clean cut. Don't keep sawing so you have different incisions. Okay, so hold on to the brain, make one nice long cut, and even if the blade is dull, you will feel ease into it and it'll naturally cut. Just don't be violent, just go slow, nice and steady. And then once you get to the cerebellum, continue the same straight line. Cut straight through, like cutting the cerebellum in exactly 1/2, and then same thing with the spinal cord, which is a little bit more rubbery, so it'll take a little more effort. So what I do is I turn, so I take the tip of my blade, and voila. The tip of my blade is sharper, so that's how I end up cutting it nicely. And nice beautiful cut here, as you can see. Take a look at the brain, sagittal cut. And what I'm going to do now is show you all the parts. So first things first, what you're going to see over here, that would be the corpus callosum, third ventricle, fourth ventricle here. What else? Beyond the corpus callosum, you're going to want to take a look and find the hypothalamus. So the hypothalamus should be below the thalamus. So if this is the corpus callosum, that's the thalamus region. Hypothalamus, pituitary gland, mammillary body right here. And then you're going to take a look and say, where is the midbrain, pons, and medulla? So midbrain, pons, medulla. One more time, midbrain, pons, medulla, and then this is the spinal cord. This is your cerebellum, looks like a cauliflower. And then inside you can see the white matter in the shape of a tree. So we call that arbe or arbor, A-R-B-O-R, arbor vitae right here. So it's the white matter inside the cerebellum, which has to do with posture and balance and proprioception. And so the white matter is in the form of a tree, and we literally name that tree of life. In Latin, it's arbor vitae. All right, so again, that's pretty much it. You need to be able to identify the corpus callosum, the thalamus, the hypothalamus. The fourth ventricle should be right adjacent to the cerebellum. The third ventricle is up here. You can see the cerebral aqueduct. And don't forget, you really should be able to identify midbrain, pons, medulla, and then spinal cord. So just follow it by order. So here's the cerebellum, midbrain, pons, medulla, big bulge here, and then straight line. It's a structure. The spinal cord is nice and straight, just bulges here, indicating the midbrain, pons, and medulla. All right, and that's pretty much it. I hope you enjoyed this video, and I'll see you in lab, ready to